Hi, editing human DNA just got a lot easier. We have been using CRISPR technology for 13 years, which mainly cuts out parts of DNA. But it is quite hard to use and it is not that much effective and it is not that much accurate. It is used as a cure in some specific diseases, but you have to directly introduce the CRISPR proteins to the cells. And you need cells to start to replicate. Some cells in our bodies do not replicate that much. And some of them has extra defenses for DNA changes like our nerve cells and brain cells. But MIT scientists figured out a new way to do this. It is much more effective and accurate. But for you to get the difference in the new technology, I'm just going to talk about the old way we are doing things. So before everything happened, the universe started and we figured out that bacteria are protecting themselves from viruses with a specific protein. This protein can wander around in the cells and can detect specific RNA or DNA sequences to cut them open. So if a virus the bacteria does not want comes in contact with our bacteria, this protein just protects our bacteria by cutting open those viruses, specifically their DNA or RNA. After finding out this protein, we didn't immediately started changing our human DNA, but along the way we figured out new things, and in 2012 we made the first DNA cut in the labs. So with CRISPR you can just delete a part of the DNA and then the cell can fix the rest of the DNA, glue them together. But this sometimes does not work. Sometimes you can cut open the wrong places. The CRISPR can start from a delayed DNA sequence. It's probably not a problem when you are trying to kill the viruses in the bacteria, but it is obviously not made for precision in human DNA. So what MIT scientists figured out is we don't have to cut open all of the DNA. The DNA have two strands. With the prime editing technology, you can just start from wherever you want. You are opening the two strands of it, you remove one strand of it, and you put the new sequence you want there. And into the original strand you have there, you are putting some small false signals that tells that this is a new strand of DNA. So you are actually lying about this being the new one, and your injected DNA being the old one. Then in a regular DNA check, the cell finds out that these are not matching. Because you put in a new sequence there, they have to exactly match and they are not matching now. So our cell's autocorrection system kicks in. When the two DNA strands don't match, the cell picks the old one. It is obviously the original one, right? But you lied about it. So the cell now deletes the unwanted part and completely fixates your new DNA strand and makes it a permanent one. The actual advantage of this method is you are not cutting open the DNA now. You are just separating the strands and changing them. So the full DNA is actually still connected from start to end. This reduced the overall error rates in the CRISPR technology from 15% to 1% and in some cases 0.2%. We are really getting closer to actually changing our DNA. Before making you scared about some company changing your DNA, we still have lots of things to do. Like, this is not easy to put into your body. These are still proteins and our body defense systems are really great at destroying strange proteins. So you still have to work harder to make this work. At this stage it cannot be something like a airborne disease that can spread and cause an apocalypse. So let's just forget about the negative scenarios, okay? When it comes to positive scenarios, we can really fix diseases. The genetic diseases that we shouldn't be able to fix actually, but we can do it. We are all born with a specific DNA sequence. We cannot change our genes, but we can now. Like, not actually now, but we are going to be able to do it in the upcoming years. For now, I'm just happy that there's a hope for all the people who are struggling with genetic diseases. In school you are being told that some diseases cannot be fixed, but we are going to be able to fix them. We still have to figure out what part of the DNA does what actually. With the Human Genome Project, we have all of the DNA in our hands now, so we know all of the code, but we just don't know what that does. So there's lots of research to do. You just don't want to change something that is going to cause more problems. And obviously I skipped lots of parts here. If you are really curious about this, go ahead and read the research papers yourself and tell me what you think about a world where we can change our DNA. It is obviously too much power for humanity right now, but I'm curious about what you think the outcomes are going to be. So let me know what you think in the comments down below. Also, while we are still humans here, like no AI in between us and not those genetically changed Barbies and Kens, am I right? While we still exist in the world, go ahead and check out my games, please. I made a fun and kinda hard counting ship to sleep game. You are making the ship jump, but there are multiple ways to play it. It gets harder and harder, but if it's not for you, I made a number grouping puzzle game. It is a good brain exercise. Go ahead and try it on Google Play and Apple App Store. I would really like your feedback on them, but it's fine if you don't want to do that. If you liked the video, please subscribe and thank you for listening.